For the second part of our tissue video, we're going to pick up with nerve cell physiology. Now, nerve cell physiology is going to be very important in how the nerves work. So we want to go ahead and talk about nerve cell physiology and how important it is so you can understand how nerves communicate with other nerves and how venoms such as toxins and poisons and drugs can actually work on the nervous system. So let's start talking about things such as resting membrane potential. So let us start off here. If you take this little stick here, this electrode, and you stick it into a dendrite or an axon or even into the nerve cell or the neuron cell, you can actually pick up the voltage that's going through here. So you stick the little electrodes into one of these areas and then you attach it to an oscilloscope, and that's going to help you to measure the voltage that's in that cell. And so when you do this, you can look over to the side and you notice that this cell is going to be at a negative 70 when it is resting. So its resting membrane potential is going to be at negative 70 millivolts in this particular cell that you're looking at. So we have the nerve cells and the cells in the body that have the ability to maintain the voltage on the interior of that cell, and they have to do that as we'll describe in a little bit in the future slides. They can do this through little small channels that they're going to have that do different jobs to help to regulate this voltage on the interior so that that resting membrane potential is going to be regulated at the negative 70 millivolts, and it's going to be accomplished by these little channels here that we're getting ready to see in the next picture here. So here with these channels, these are going to be found inside the membrane, and so your cells can use these as players that are going to be the ones that are regulating the sodium-potassium pumps and the potassium channels and sodium channels, and they're going to help with leak channels and things such as that. So with these different sodium-potassium channels, we will call these leak channels. And so what's happening here, the first one is going to be the sodium-potassium pump. So for every 3 sodiums that you see here that are going to be pumped out of the cell, the bottom, you're going to see that there's going to be 2 potassiums that are going to be pumped into the cell. And so that's what this sodium-potassium pump is going to do. This sodium-potassium pump is going to be really important in maintaining the balance of ions in your cells of your body. If you notice, sodium is more prevalent on the outside of the nerve cells than it is on the inside of these cells. And so we have much more sodium outside than we have inside. So another way of stating that is that the extracellular environment is going to have more sodium and the intercellular environment is going to have more potassium. And so looking at that, you can see that from this diagram. So also notice how you're going to have more potassium leak channels than you have sodium leak channels. So we're going to have these leak channels being able to leak more positive ions out than the negative ions that are going to be coming in. You can also notice that you're going to have more positive charges leaving than you have positive charges that are coming in. So as more positives leak out, of course, that's going to help to make the inside more negative. Also, when we're looking over at that sodium-potassium pump, the one that's there in the yellowy orange color over to the left-hand side, notice that for every 3 positives that it pushes out of the sodium, it's only bringing in 2 positives. So that also is going to help to keep that negative state there at negative 70 millivolts. So this negative 70 millivolts is going to be a common measure or a common voltage found in all cells. This is going to help to regulate things such as water coming in and out and other ions that are coming in and out without the cells. So the resting membrane potential for most cells is going to be negative 70 millivolts. And for note purposes for note 6, this just goes over that. So inside the cell, there's going to be more potassium, which we just saw. Then there was sodium. And the outside of the cell is going to be the opposite of that. So we said there were more potassium leak channels than there were calcium leak channels in that cell membrane. So more positives will leave the cell than that come in. Also, what we looked at was that for every 3 positive charges from potassium that's going to leak out, there's only going to be 2 positives that's going to come in of the calcium that's leaking in. So that's going to help keep that negative charge there for you. So here's a video that you can watch on your own. This is going to be in the PowerPoint. This goes over it, kind of shows you a diagram, or goes through it and lets you actually see a visual of what's happening with these different leak channels. So there's going to be different types of channels also that's going to allow different ions in. Here we see you have a chemically gated ion channel. Here it's going to be open in response to binding of the appropriate neurotransmitters. So you have a receptor that's going to be found on that protein that's going to be embedded in that cell membrane. So what happens is that receptor is going to be open until a neurotransmitter comes in and binds. When that neurotransmitter that's a chemical comes in and attaches to that receptor, notice how it opens up that channel. So the channel here was closed. It's going to open up that channel here, and it's going to allow it to send ions through. And so you can see that potassium is going to leave. So K is going to be your potassium. It's coming out. And sodium, which is Na, is going to be coming into the cell. And so because that chemical bonds and adds it to that, it's going to allow that opening and that closing. Once the neurotransmitter breaks apart, usually there's going to be some type of enzyme that comes in and it will break down that neurotransmitter, cause it to release from that activation site or that receptor site. It will then close back again and won't allow any of those ions to come through. Another way we can control the charge of the cell is by the change in voltage. So for example, normally it's going to be negative. We see inside of the cell here, and it is positive on the outside. This is going to keep ions from coming through. But if for some reason the inside changes to a positive charge, it will cause this other type of protein, which is called a voltage-gated ion channel, which is going to be found embedded within the membrane. It will actually change, and see negative on the outside, it's actually going to change the shape of that protein. And when it changes the shape of that protein, it's going to allow that channel to open up to allow ions to come through. So we can stimulate with drugs or other kinds of venoms and toxins. We can stimulate the membrane to depolarize. That has become more positive. We can use drugs and things to stimulate the resting membrane potential to be hyperpolarized, like in the second picture there, and become more negative. So this is how drugs work and how venoms and toxins work. They cause depolarizations to occur, or they can cause hyperpolarizations to occur and change that membrane's resting potential from negative 70 millivolts to whatever it needs to change it to. There are going to be some different types of potentials. You can have graded potentials. These are going to be short-lived, localized changes in membrane potential. These are going to be stronger. The stronger the stimulus, the more the inner membrane voltage changes. Graded potentials will occur on the neuron cell body. And so looking here, you can see we have membrane potentials. And so as it comes through and depolarizes in a region, you're touching it with the stimulus there, you're depolarizing it in that section. And you can see that that small patch of that membrane in red is that depolarization station. Now, depolarization can spread, so that opposite charge can attract each other. And that's going to create local currents. That's those black arrows that you see there that depolarize the adjacent membrane areas, and it's going to spread the wave of depolarization down that axon. So membrane potential decays with distance. So once it goes from that spot where it was touched, it starts to depolarize the sections near it, but as it moves out, because that current is lost through those leaky plasma membranes, the voltage is going to decline with distance from the stimulus as it moves down. So you see how it starts to decline on both sides as it goes away from the main point of stimulus. Small deviations in resting membrane potential could happen. So we have over here, the first one, you have what's called a hyperpolarization graded potential. You can see how it goes down below that negative 70 millivolts. Or you could have depolarization graded potential, where that resting membrane is going to start off, but then it goes and it becomes closer to, let's say, negative 60 millivolts. So we know some positives are leaking in there. With the graded potentials, it's going to occur in response to the opening of a mechanically gated or ligand-gated ion channel. So, different ways that we could do these different graded potentials. You could have what's known as a mechanically gated channel, and that is going to be where you can apply a pressure. When you apply pressure to it, it will then go in and it will open this channel up, and it's going to allow those ions to flow through. Another way you can have it is by using a neurotransmitter to depolarize. And so with the neurotransmitter, it's going to come in, it's going to attach to that receptor site. Once it attaches to that receptor site, you see how it opens up the channel and allows the flow back and forth of these different ions. And that's going to help with a ligand stimulus. And both of these are going to be depolarizing. Now you can hyperpolarize like what we just saw in the last chart, and you can do that by going in and you can put certain types of neurotransmitters or medications, things like that, into these different receptors, and they can actually pull in negative charges instead of positive charges. And so that would hyperpolarize that potential. And so amplitude of a graded potential is going to depend on the stimulus strength. So you come in, you have your resting membrane at negative 70 millivolts here. And so stimulus 1 comes in. Notice how if you look at the bottom, it's not as strong. You see a little rise in depolarization. You come in with stimulus 2 a little bit later after it repolarized. You see it's a little bit stronger, and that will measure on the chart here a little bit stronger. And at the bottom, when you're sitting here looking at how strong stimulus 3 is after it repolarized, you're going to see how that made an even higher move towards depolarization. And then it goes back to its resting membrane. Graded potentials can be added together, and when they do that, this is going to make a larger amplitude. So that's going to be referred to or called a summation. And so you send stimulus 1 in, and before it can come down and completely repolarize, a stimulus 2 comes in. And so you see a little small dip, and as stimulus 2 comes in, it's going to come in and it's going to add to that stimulus 1, and it's going to make it a higher or larger amplitude, and then it returns to its resting membrane potential. Action potentials. These are going to be a strong depolarization, repolarization, and hyperpolarization of that inner membrane. It's going to occur along the axon and muscle cell membranes. Action potentials are going to be transmitted for long distances across cell membranes. And so here you see you're going to come in, you have your first action potential that's going to be working on the axon of that neuron there. And so we have our depolarization, so we see the positive particles coming in. It's going to start to move its way down as it depolarizes. So we depolarized here. As it moves its way down, it's going to work on a repolarization and then goes back to its resting membrane potential at the bottom. So you can see how it moves down that axon. Steps to the production of an action potential. First step, you're going to have a resting state, and that's going to be where sodium channels and potassium channels are going to be closed. And so that's what you see in part 1 there. The second part, you're going to have what's known as depolarization. That's what we've been talking about. A stimulus is going to come in. If it's strong enough, it's going to cause voltage-gated sodium channels to open. Sodium is going to begin to diffuse into the cell. The third step that you see on there is going to be the rising of action potential. So that's where the sodium channels are going to allow sodium to flow in, making the inside of the membrane positive with respect to the outside. Step 4, you're going to have that falling of action potential. And so the sodium channels are going to close and potassium channels are going to open. This is going to cause the inside to become negative. Usually after that, you have what's known as an undershoot. So those potassium channels are going to remain open and cause the inside of the cell to become more negative than the resting potential. So potassium channels eventually close, and that allows the cell to go back to its resting potential state of that negative 70 millivolts. So action potential is going to be caused by permeability changes in that plasma membrane. And so here's the different what's coming in. So right around the step 2, you can see how sodium permeability is going to be the highest. And then as you're coming down off of step 3 to step 4, you're going to see that potassium permeability is going to be at its highest. So coming in here, we have our first action. This is just going through this again with these potential resting states and sodium channels and showing you how everything works. And so first we have our resting state. And so that channel is going to be closed. No sodium, no potassium are coming in. So that's what's happening here. Next, you have a threshold. So you're going to have a stimulus coming in. You have the activation gate that's going to open for the sodium, allow it to come in. And you see how all of this is flowing in. So remember, sodium is going to start to come in first. Depolarization phase of action potential. So that's when we're going to be at this third stage. You still have your sodium coming in. Then the repolarization phase is going to occur. And so you have these sodium gates and channels close. And now you see these potassium channels open, which are going to allow that potassium to leak out. Then a little bit too much will leak out at first. And so you have that undershoot, and then it will bounce back up to that resting potential of negative 70 millivolts. And here's just another visual of what we went over. So you have your resting state, everything's closed. Depolarization allows that sodium to leak in. Repolarization, you're going to close the sodium channel, open the potassium channel. You allow that potassium to leak out. You have a slight hyperpolarization, and then you return to the resting state. Okay, and so this is what it looks like as it's going to be traveling down the axon. So at 0 milliseconds, you see the action potential has not yet reached the recording electrode. By 2 milliseconds, action potential is going to peak, and it reaches that recording electrode. At 4 milliseconds, you're going to see the action potential peak has passed that recording electrode, and the membrane at the recording electrode is going to still be hyperpolarized. But if you look right ahead of it, you're going to see that it's starting to return to its resting potential. So this chart shows you 2 different stimulus. One's going to be a lot weaker than the other one. So if you look at it, notice how the first one is going to be a slight stimulus. You can see those action potentials that are being activated there. When you come down a little bit further, you see a much stronger stimulus coming in, and you can see that there in those action potentials. So in the bare plasma membrane, you're going to see voltage start to decay. So right there at the top with the stimulus, right here, you see that's where it's going to be the strongest. As it travels down that axon, it's going to start to get weaker and weaker, and the action potential actually decays. So it's not going to be as strong. So this is going to be traveling actually down the dendrite here. In the non-myelinated axons, the conduction is going to be slow. They could call it like a continuous conduction. So here we're going to have voltage-gated sodium and potassium channels that are going to regenerate that action potential at each point along the axon. So voltage doesn't decay. And so you will see that it starts out strong here, travels, starts to decay slightly, and then it picks up again, sends it through, and it slightly decays and then keeps re-establishing that voltage. So this keeps it from decaying completely. So conduction is going to be slow because it takes time for these ions and these gates and these channels to open up and to move these ions in and out and to send the signal down. In a myelinated axon, however, conduction is going to be very fast. And so this you can think of as a saltatory conduction. Myelin is going to keep the current in the axons so that voltage doesn't decay, and is going to help it to travel much faster. So the action potential is going to be generated only in the myelin sheath, and the gaps are going to appear where it kind of jumps across, and then it reestablishes its right through here, it's going to reestablish its action potential and then travel down. So the top one, think of running very, very slow, maybe a runner or a sprinter. The bottom one, think of being able to get in a car and go 100, 200 miles an hour. So it's going to be extremely different. So myelinated sheaths are going to be able to allow you to send that signal down and allow it to travel much further without decay. And so here you see that cell body. The action potential is going to actually propagate and start to change over into the negative 70 millivolts right here towards the end of that cell body and start to travel down through that axon. Now, that Schwann cell is what you're going to have through all of your peripheral nervous system tissue, all of your nerves that are going to be traveling down through, let's see, your arms, your appendages, through your body. Remember, if it is going to be in the brain or the central nervous system, it is going to be an oligodendrocyte that's going to be myelinating these axons. So that's the two different ones. You have your Schwann cell and the oligodendrocytes, and the difference that you need to know, the Schwann cells are going to be all over the body. The oligodendrocytes are going to be the ones that are going to be around, like the neurons in the brain. And so this shows you how they're traveling down this myelinated sheath. And here's another visual just to help you. It shows the first step, that action potential is going to occur at an initial segment. So you see it coming in. The local current produces a graded depolarization that's going to bring the axon membrane at the next node to the threshold. Remember, the axon membrane is going to be called an axolemma, kind of like the muscle cell membrane was called a sarcolemma. Third step, that action potential is going to develop at node 2. Fourth step, you have a local current that produces a graded depolarization that's going to bring the axolemma at node 3 to its threshold. And so you can see how fast it's going to travel. At 1 millisecond, we see how that at the axon hillock, it's taking off. The current flow, due to the opening of sodium channels, is going to make its way down that axon. By 5 milliseconds, you see how it has traveled. And by 10 milliseconds, you can see how it's gotten about maybe a fourth of the way down. Now coming over to the saltatory conduction or the myelinated axons, and you can see a difference. So you see at the top one at 1 millisecond, you start to see that action potential start at the axon hillock. By 5 milliseconds, it's made its way down a little bit further. And by 10 milliseconds, it's made its way all the way down almost to its axon terminals, where it will attach to a tissue, a gland, or another neuron. So synapses between neurons, you can see how neurons can be attached to many, many other different neurons or to different types of tissues. So you see that you're going to have one that's going to come in and it's going to attach right there at that axon hillock. This one that attaches at that axon hillock is going to send information into that area there. And so that's called an axononic synapse. The next one, you have an axodendritic attachment, and those are going to be axons that come in. They attach to these dendrites and they send information in through the dendrites into the cell body. The next type, you have what's called an axosomatic. That's going to be the ones that come in and they're actually going to attach in on the cell body of that neuron, and then they will send their action potential down through that cell there. So that's going to be 3 types of synapses in neurons. Now when they come in, remember, you've seen this picture before with muscle tissue and how muscle tissue works. You are going to have the end portion of that axon. That's going to be your axon terminal. And it also can be referred to as the presynaptic neuron, where it's going to come in. And so remember, these two parts don't actually touch. There's going to be the opening between it called the synaptic cleft. And so you have this end terminal plate, which is going to have all of your neurotransmitter, which you see in these little vesicles. That's going to be red. You see calcium coming in through these leak channels. That calcium is actually going to help these vesicles release what they have in them. Those vesicles will hold some type of neurotransmitter. For the sake of, let's say, muscles, because we went over that in detail with muscles. Let's say it's acetylcholine. If that is acetylcholine, that calcium comes in, causes these vesicles to release that acetylcholine. Sorry, thought I had my drawing tool up. So it causes these vesicles to release the acetylcholine. That acetylcholine makes its way to this membrane where it's going to be pushed out through an exocytosis. It's going to cross the synaptic cleft here, come in, attach to these protein channels here. Once it attaches, it's going to cause those protein channels to open. When the protein channels open, it's going to allow the sodium to flow into that next tissue, and that's the postsynaptic potential. And then that's going to cause that nerve impulse to continue to travel on through that next tissue, cell, gland. All right, here we have excitatory postsynaptic potential, or EPSP. Here you have a local depolarization of that postsynaptic membrane that's going to bring the neuron closer to action potential thresholds. So that neurotransmitter binding opens the chemically gated ion channels and allows things like sodium and potassium to pass through simultaneously. So it comes through at the same time. The next one you have is going to be an inhibitory postsynaptic potential, or an IPSP. With these, you have a local hyperpolarization of the postsynaptic membrane, and that's going to drive the neuron away from the action potential threshold, and the neurotransmitter binding opens the potassium and chlorine channels. And so notice that chlorine is going to have a negative charge, and so that's going to send that stimulus the opposite direction and hyperpolarize it. Now with neurons, they are going to be attached to many other neurons. And so lots of times you're going to have different neurons acting on each other, acting against each other. And so here you can see all different types of neurons that are attaching to each other. And so you have different things like excitatory synapses and inhibitory synapses. And so in order to have an action potential come through or have a neuron respond, you're going to need to have more excitatory, if you want it to actually send action potential out, than inhibitory. And so coming in, you can see here with this specific neuron, you're going to have 5 different presynaptic neurons that are going to attach and come in and send responses in. And so you have 3 that are going to be excitatory neurotransmitters that are coming in. You're going to have 2 that are going to be inhibiting neurotransmitters that are coming in. And so with that, since you have more excitatory, they're going to be able to stimulate that trigger zone. And since you have more action potential coming in through there, it's going to be able to travel down that postsynaptic neuron. And so here it's going to be able to show you different neurotransmitters, and you can read about different things that they're going to be able to control and their locations. So if it's a CNS, it's going to be in the central nervous system, so that will consist of the brain and the spinal cord. If it's going to be the PNS, or the peripheral nervous system, that will be anything that comes off that spinal cord. So that's going to be the peripheral system. That's going to be those nerves that come off the spine and go down the arms, down the legs, go through the face, the brain, the head, places like that around the trunk. Right. Looking at this is going to be a chemically gated ion channel. So you have your acetylcholine, which is going to be your neurotransmitter. It's going to come in, it binds to that binding site, and it's going to cause the opening of that channel. Once that channel opens, you can see that sodium, which is a positive ion, is going to be able to come in, and it's going to be able to make its way into that cell. So that's a direct effect. Indirect effect. We're going to have that neurotransmitter come in. It goes to that G protein. Once it attaches there, that G protein becomes active. You're going to have ATP within that cell that's going to mix with the adenylate cyclase. That's going to be an enzyme that acts as a second messenger. That's going to be called a CAMP. That in turn goes back over and opens up that ion channel. And so this is how we're going to be able to open this one. So that's going to be an indirect opening using a G protein here. And so this one does require ATP or some type of energy. Indirect effects by intracellular enzymes are going to be things like nitric oxide and carbon monoxide, things such as that. And so what happens is you'll have these gases like nitrous oxide, is the one that they're using here in this diagram. They're able to diffuse directly across the membrane. They don't need a protein channel or anything to help them get across. Once they diffuse into the cell, it's going to activate enzymes. These enzymes are going to produce a second messenger. The second messenger comes over and it will then open that ion gated or that channel that's going to allow the ions to come in. And so that's going to change the cell's metabolism and activity using these. Next, we have neural circuits. A neural circuit is going to be a functional group of neurons that process specific types of information. Types of circuits that you may have might be a simple series, diverging, converging, reverberating, or parallel after discharge. And so let's look at these. All right, we have the diverging. So we have an original cell that comes in that's going to have an action potential. It is going to start to diverge. It goes out to 2, then maybe go out to 4, and it can continue to go out and diverge into more and more. A convergent is where you take several and you'll bring it in to 1. And so they kind of converge and come in together. Next, with the reverberating, these are going to be where it comes in from a cell. It might be attaching going straight through a straight line, but you might have some off to the side where it can come back and reattach in to that same cell and send the signal down. So they keep coming back and they can make little circles and little loops, make their way back around, but yet continue to go down. With parallel, these are going to be able to start off and then to branch out into parallel, just like what it says, and have parallel units or circuits going down, and then all can come in and attach to one at the end. Although the nervous system exhibits plasticity, that means it's able to change, it's able to rewire itself, neurons are going to have a limited ability to regenerate themselves. So lots of times you hear that if you lose brain cells, it's for good. They do not go through mitosis, they don't regenerate themselves, and for the most part, they don't repair themselves if the damage is really bad. So plasticity, just going over that, the capability to change based on experience, so rewire themselves, learn things, regenerates the ability to replicate themselves. Now with this, they found out that this isn't exactly true. Now if you lose 100 brain cells by hitting your head, you're not going to be able to replace those. But sometimes they do go through mitosis and they do replace themselves. But it could take anywhere from 10 to 40 years for these cells to replace themselves. So that's why we tend to say that they don't really regenerate themselves, because they can't repair themselves or they can't replace themselves in the numbers that needed to be replaced as you lose them. So in the central nervous system, there's going to be very little repair. That's going to be in the brain and in the spinal cord. But in the peripheral nervous system, repairs are going to be possible as long as the cell body is intact. So the cell body is going to be the main portion of the neuron. And so that's going to be this section here. It's going to have things such as Nissl bodies, which are going to be there to help with it. So this on top is going to be your normal neuron. Coming down, we see that we have what's called a chromatolysis, and a Wallerian degeneration. And so what happens there, as long as your cell body is still there and you have some of these Schwann cells that are still able to come in, it's able to go in. They can start to repair that axon there, and it creates what's known as a regeneration tube. The axon then is able to be repaired, put back together, and then you're going to have these Schwann cells start to form and wrap themselves back around that axon. With this is going to be a video. It's a good video that kind of goes through and talks about some interesting things with the nervous system. And I strongly suggest that you watch this from the PowerPoint. And that's going to finish us up for our second part of nervous tissue.